Okay, thank you very much. And indeed, I'm gonna present to you an update, or maybe it's the first time you hear about the European Research Initiative on CLL, also named ERIC. I had a couple of questions to ask you through the app, but since we are just a few friends, I'm not gonna bother you asking you to open the app and respond. The first one was just to have an idea from which area of the world you are coming, but now we are too few, probably those who are closer and they don't need an airplane to go home. And so we just keep it, thank you. They are not gonna vote, so if you can proceed with the next question, which instead it is, so if you want to vote, vote. So, 0, very good. Everybody from, so John Bird is the only one left in. So the next question is indeed if you know about ERIC, so we can just have a quick poll in this very moment. Who is a member of the ERIC? You can raise your hand. I see, so the ERIC members are very faithful and they remain, I would say 1/3 maybe. I just want to be optimistic. And, but it's okay, very good. Thank you for staying. So we can move also on with this slide. So as you will see, ERIC is also officially, thank you. Oh, they voted, very nice. So ERIC is a scientific community of scientists with a passion for CLL research. So that's the key point from where ERIC started and where ERIC is going. And it is also a German non-profit organization, just because Michael Alec in his spare time likes to go around and found non-profit associations. So, and that was indeed founded by Michael Alec and I think Emily Montserrat and Guillaume Di Ghiero, but I don't want to make mistakes, I was not there at that time, in 2006, and always for legal administrative reasons. The executive office, thanks to Emily Montserrat a few years back, is located in Barcelona. And we are also recognized at the European level from the European community, so we can apply to European grants. And actually, the ERIC is born as a byproduct, I would say, of the European Leukemia Net, since we were originally the work package number 7 that was dedicated to CLL, and then from that organization, ERIC stemmed out. We are also the working group of EHA, again dedicated to CLL. So somehow in Europe, whenever you talk about CLL, you talk about ERIC in all these situations, just for the simple reason that ERIC is not something top-down, but something bottom-up, meaning that it's made by the people who are interested in CLL, and they join and they work together, so without any pressure from above, from any particular board of thinkers. So as you can see here, it is indeed collecting 1,296 members as of today from 79 countries. So you understand that the borders already stretched far beyond Europe, 680 different institutions. And as you can see, from 2015, we really started increasing the number of members. And I think that if you join those who are not members yet, I mean, we will increase even more. So just to show you, it's really covering the whole 5 continents. And I have to say, in the last couple of years, we really started also getting to Africa because there is, again, also there interest in CLL. We already heard about Asia, India, China, and so on. So what is our mission? The mission is to improve the outcome of patients with CLL through research, whatever type of research, basic, translational, and clinical. Of course, with the aim always of translating every basic finding into clinical application. You will see that that became really part of our mission. And that is the major idea is that this can be done only at collaborative level. So this is just to make you understand that Eric is not trying to do research in contrast, in competition with the single investigator, single institution, single countries. Whenever an institution, an investigator, a local network, a national network feels that they can achieve whatever they aim in their research alone, it's fine. Whenever you feel that you need more resources, more patients, more biological material, then Eric is there to help as a resource for everyone. And so now we are not yet anymore fostering collaboration in Europe only, but at international level, because that's what happens every day. I mean, friends and colleagues are all over the world, and of course we don't put any limit who is collaborating and entering the projects. Again, one important thing is that there are not Eric projects, meaning that there is not Eric who is given a project and either you like it or not. There is always a member of the Eric who is proposing a project. So someone who had an idea, he feels that he cannot achieve that alone, and then he needs just more blood, more cases, more patients, whatever, and then he asks Eric to inform the members about this project, and whoever is willing and whoever likes that project can join and make the project. So I just show you a few examples. I don't go into the scientific details. Some of them are published. You can read. Some others will hopefully be published. This is one study where, for example, was proposed by the Italian colleagues. Sorry, I start with this one for the conflict of interest. They wanted to understand the efficacy and safety of bendamustine plus rituximab only in second line, because that's probably the only niche that remains for bendamustine unless you use it in first line. The paper was published in Hematologica, 35 different institutions, not only Italians, of course, but all around the world. There has been a spin-off of this project, and now we have a collaboration with Anthony Mato. You will be hearing later speaking. Because we wanted to compare then the results of the bendamustine in second line with ibrutinib treated patients, and therefore they joined the project with 70 data from 70 institutions, and this is something that is ongoing, and you will be hearing and very likely published in the next month. Another study, for example, was proposed by Yair Herishanu, so from the Israeli CLL study group. They wanted to examine the efficacy and safety of obinutuzumab plus or chlorambucil, or even obinutuzumab alone. In some countries, people are using it. And therefore, we put together 460 patients from 51 institutions in 13 different countries that you can see listed there beyond, of course, Israel. Another project that you heard yesterday presented by Leslie Sutton and made in collaboration with Lydia Scarfo and Silvia Bolfiglio and many other colleagues from 20 institutions in 10 different countries is the story about the presence and the frequency of BTK or PLC gamma 2 mutations in patients becoming resistant to ibrutinib. And what we showed, and now we confirm, is that roughly only 50% of the patients do develop a mutation in BTK. So thus, another 50% must have a different mechanism, which is not PLC gamma 2, and therefore this also needs more research. Another important finding, I think, that you heard on the hot topic of cytogenetics, and in particular complex karyotype, Panos Baljakas from Uppsala, together with another 17 medical centers all around the world, collected over 5,000 cases of chromosome binding and to really understand what is complex karyotype in CLL, and therefore he published a paper in Blood this year, then really showing that the threshold to consider complex karyotype is 5 or more aberration, not the usual and typical 3. So we have somehow to revisit all the data that we might have in mind, all the previous publication, and see, especially in prospective studies, if this holds true or not. One last thing, but last but least, as I said, we can apply for European grant, and indeed we are a partner in this European grant funded by IMI. Innovative Medicine Initiative, which is a shared initiative between public institutions, so academia, and private institutions, so pharma companies. And within this consortium, who is now funded by the European Community, the ERIC is a partner and is coordinating, of course, the project on CLL, though the project is covering many other hematologic malignancies, as you can see from the title, Big Data to Enable Better and Faster Treatments for Patients with Hematological Malignancies. They are the project coordinated by Leslie Sutton and Anastasia Dimitriou. Is aiming to evaluate 11 of the most frequently mutated genes in CLL, which are not so frequently mutated at the end, so they are mutated only in a small proportion of patients, so you need to put together a large number of patients in order to assess the prognostic and clinical relevance. Almost 5,000 patients put together from 27 institutions in 12 different countries. Something new, which I think is, will really open a new, let's say, topic, a new field for us as ERIC, is a patient empowerment program. This is not correct, sorry. This is the database, sorry, the title is wrong. So we launched the collection of data within a database that can be shared with all the institutions that are willing to have it. And now we collected so far more than 5,000 patients from 11 centers, 6 countries, and it has been launched at THA, and every institution can join the ERIC database or can use the ERIC database for its own local purposes. It's a very minimal, what we call minimal database, to collect the minimal information, but also in order to know how many patients you have, for how many patients you have collected biological material and is available. And of course, this is the basis for all the future projects within ERIC. We also are planning to do it prospectively, so you can use that tool to collect your patient and keep the information. And maybe in the future, it will be linked to the electronic medical records that you all have or might have in the future in your institution. So as I said, a new line of activity, but also research, is collaboration with the patients' organization, with the Patient Empowerment Program. And indeed, for the first time this year, we were part of the patients' organization meeting, which took place on Thursday and Friday here in Edinburgh. And we are really designing a program where we are assessing the needs of both the patients, but also the doctors, and in order to make them talk and relate in a more effective way. So, this, of course, we are still academic people, and therefore everything we do, we research, at the end we want to have it published, and that's something, an outcome that is always at the end of our projects. So this is just the most recent publication in the past 12 months, but ERIC published even in the past year. And you can see one is the paper on cytogenetic complexity, the other one is the paper with on BR and so on. The other thing that, of course, what is really our nature, probably, the type of research that we are doing is becoming, in some cases, rather technical, I would say. So we are really keen on understanding how certain basic information can be applied and transferred to the clinic, and how that can become a tool that the physician can use to better manage the patients. So this made us, of course, becoming interested in particular molecules. The first one that has been a failure, a total failure, the first one was ZAP70, meaning that, of course, we wanted to standardize the test for ZAP70, so that could be reproducible, used in clinical trials and multi-center clinical trials, and that was never possible. So that's the reason why I think very few of you, if any, probably is using ZAP70 in clinical activity or in clinical trials, because it's very difficult to reproduce. If you do the analysis today or tomorrow, if you do it today or in 2 hours, it will be always a little bit different. So it's really technically impossible to standardize it. Much more success we had with minimal residual disease analysis. You know, the test for flow cytometry is now the gold standard, recognized by FDA and EMA. With flow, of course, immunoglobulin gene analysis, I don't have to talk to you about that. You heard the Kostas Stamatopoulos. We wrote the guidelines that maybe and hopefully many of you are following how to do a reliable analysis on immunoglobulin. Same thing holds true for P53 analysis, thanks to Stefan Stingelbauer and Shalka Pospisilova. And now we are moving to novel gene mutations, and in particular also the complex karyotype, hoping to really define what can be the best strategy and assay to assess complex karyotype. Yesterday we had a discussion about the arrays, and we will see what's the best. So that's the reason why we moved a little bit further. So for the analyses that are really recognized as necessary for the management of our patient, we created networks of laboratories that are reference for all those who want to set up the analysis and have advice and help. And we also created a certification procedure so that all laboratories can apply, and therefore they can be certified to be reassured to provide a reliable analysis to their patient. That is true for P53. 167 laboratories all around the world are certified at the moment, and they are in Europe plus Australia, Brazil, Canada, and India. Same thing also for immunoglobulin gene analysis. We have 92 centers at the moment. We started the process a little bit later compared to P53. Again, all over Europe, plus Argentina, Australia, Taiwan, and USA. And since it's becoming such a huge effort, so the ERIC will continue certify initially every lab who will make a request. It's all for free. It's all provided by ERIC. We just want to be sure that the laboratories that are providing the analysis are really providing a reliable value. But for the recertification of the laboratories that have been already certified initially, then we join a collaboration with UK NECAS that probably many of you know, because it's certifying laboratories for many other technologies all around the world. And that will start really this autumn with this pilot study, and then officially in January 2020. So the last thing, since we publish guidelines, we think we set up standards and techniques that can be, should be used for the management of our patient. We also like to educate the community about this and about the relevance of performing this test. So the next point is meetings and educational events, which are very specific on these particular techniques. We started many years ago, now it's 12 years ago, with a workshop on immunoglobulin gene analysis. These are very practical workshops, so meaning not really working on a bench, of course. It's always a nice place in very nice places all around the world, but it's really gathering the people in small groups to discuss issues and problems and to help to set up and run the test. So we don't aim for large events. It's always events that include 50 to 100 people. Usually it's always more than 100 because people always request, so we always go above the limit that we wanted. From Europe, we moved to Middle East. In the last 2-3 years, we went to Israel, Palestine, and then for the first year, this year, we went to the US. For P53 analysis, we had also a couple of these workshops, plus a more generic workshop on biomarkers in Russia, in Belgrade. And we started again this year for the first time a workshop on MRD analysis, which is not yet to be incorporated in the clinical assessment, but very likely in the future it will become relevant, and therefore we started educating the community. Starting 2019, all the workshops are also available online, thanks to a collaboration with VUMEDI, which is an association that is really collecting, recording the presentation, and then making it available online. 2020, we are moving forward. We are also going for P53 immunoglobulin analysis in India, in Japan for MRD analysis, and maybe we are discussing now to go back again for immunoglobulin gene analysis to US, to the Mayo Clinic in Scottsdale. What we are very proud, thanks again also to Emilie Montserrat and Carol Moreno in Barcelona, we organized the first international meeting, a larger meeting in 2018, but again, not a huge event with a close number of people, because again, what is our ideal meeting is to gather people in small groups. So a maximum of 300 attendees, the rest was faculty, divided. They were then split in 4 different groups discussing about whatever we found relevant. It was how to apply immunoglobulin genes, how to manage the side effects of the novel drugs, or whatever can be relevant. Really to be practical and really with the hope to change and improve the quality of management of our patients. People from 32 countries applied, and we are going to do the same thing in 2020 again in Barcelona, which will probably will remain the city where we are going to do these meetings. And please save the date and take a note. Again, the number will be limited, so if you wish to do so, it's better you register as soon as possible. So of course, not all this can be done by only by me, but you need always good people and smart people. You can see the people around, and you know that these are probably the top experts in each of the field. So we have Andy Rostrom, Richard Rogan, Chris Costa Stamatopoulos, Salka Pospisilova, Caro Morena, Arno Kater, plus Anastasia and Leslie, who are the new generation. And in Barcelona, we have also an administrative support, which is made by Esther, who is running the office in Barcelona. I wanted to ask you again at the end, if you are planning to become a member. I hope to have 100% there. Number 3, no, but I will become soon. I hope you will do it anyhow, so it's free. You can join, just go on our website. It takes really probably 15 seconds, name, last name, institution, email, and you are a member, and you are informed about things, or you can propose projects, or you can tell us your needs, and we will try to help. Thank you very much for your attention.
Thank you, Paolo. Paolo's happy to take questions. I guess the comment would be that Eric's activities really complements the cell activities in many ways, isn't it?
Yes, indeed. That's exactly. I hope I conveyed the message. We are somehow more practical, let's say. So it's really a focus for many reasons, because probably it's also easier to foster collaboration when you have really to find a solution to a practical issue on which everybody is interested. So when we started talking 20 years ago, Nick, about immunoglobulin gene analysis, and then that became clinically relevant, everybody was using different techniques. We could not even compare the results in clinical trials. I mean, my case were mutated, your case were unmutated, who knows? So I think that is, I dream about the idea that Eric is providing a service to the community. So we are not exploiting anyone, we are not competing with probably the real research, the high-level research, but we are really providing the basis on which everyone can work reliably. So you can really run clinical trials now that are comparable everywhere all over the world. And the same thing will be true, for example, for MRD analysis, minimal residual disease analysis in the future.
I guess we did discuss at one point, I'm not sure how far it's going forward, the rare diseases that we might want to do trials across Europe or across the world. You know, Richter's is one some years ago we talked about p53 deleted cases. Is that still a possibility within the ERIC network? Yeah,
that's something that we always propose. So again, it's not Richter can be a case, but again, if you want to do a study on Notch1 mutated patients, or if you want to do patients on with blue eyes, as I always say. So I think ERIC is probably really the reservoir where that can be achieved. So if you want, like for example, Yair from Israel, he wanted to study patients treated with obinutuzumab single agent. I mean, I don't know how many people are using single agent, but there are some. So let's see. Just putting together whatever patients he found, probably 100 patients who have been treated with only obinutuzumab, so you can have some meaningful insights. So that's exactly something that we would like to do also in the future in higher number. Also prospectively.
I mean, the final comment from me is about registries, I guess, because we had a discussion about trial registries, and I know Barbara Eichhorst with her team, developed that in Germany. We've done a similar thing in the UK, which builds on the same data. But I think that what we heard of in the previous session is that in parts of the world where we don't know enough about CLL, there's a real demand to a requirement to get more information. I mean, could Eric participate in that activity?
Yes, again, Eric wants to just help and provide, again, a service. So the idea here was to provide a template, a tool with what we called at the beginning minimal data set, so the minimal information that you want to know about a CLL patient. Because the idea, again, if you ask too many information, nobody will fill up the database. If you ask the correct number of information, at least you have the picture of how many patients you have at your institution, or in Warsaw, where we are going in 2 years, or in Thessaloniki, where Costa stays. And then, when you have a project, you say, look, I want to study patients with blue eyes. Of course, in our database, we don't have the color of the eyes, but those who are interested, then they will have a motivation to add that piece of information to the database, and that could amplify the information. So I think that first of all, we should create a common tool that we all agree upon, and again, then we can use it for whatever use you want.
Okay. Paolo, thank you very much.
Thank you.
No further questions? Thanks a lot.
Thank you.